Life Science Innovation Northwest with Matt Scholz, the CEO of Immusoft. And I'm looking forward to learning about this new enterprise. I think it must have something to do with immunology. Is that a good guess?
Yeah, that's a reasonable guess.
Tell us a little bit about the company, how it got started, and then kind of where you are headed, please.
Well, I had a very interesting background. It basically approaching the immune system as though it's driven primarily by information and saying, oh, you have an immune memory, it stores that memory somewhere, so let's see if we can manipulate it. And my background is actually in computer science and physics, and we were trying to use code-breaking hardware to build new proteins, and eventually said, well, let's just build a good technology that can actually program the body to secrete whatever we'd like. And so now we turn patient cells into drug factories.
Patient cells into drug factories. Fascinating idea. Help unpack that a little bit for me, please.
Well, it is, you have all these cells and they can produce pretty much whatever you want. In the traditional medicine, we would produce a molecule and we'd inject it into a person or administer it orally, however. And in the way we look at it is we will take your cells out and say, let's just give them direct instructions on how to produce whatever the therapeutic is, and then put them back in your body and let them survive there and produce it for you. And so it's not altogether different than something like Dendrion's idea of an ex vivo vaccine, but instead of presenting your body with an antigen and hoping that it learns how to attack it, we give it direct instructions. And so we can produce things that the cells wouldn't normally produce. So instead of just antibodies, for example, we could produce lysosomal enzymes or other therapeutic proteins.
Wow, that's pretty heady stuff. So there's perhaps typically a long path from having an idea like this and seeing people's lives benefited. Where are you on that pathway?
Well, we're still relatively early. We incorporated back in 2009, and we really started on a shoestring. We got collaborators to build a prototype virus for us to program the cells, and other local collaborators to help us test it. And in the last year, we got some initial funding from the Thiel Foundation, Breakout Labs, and some local angel investors and the NIH, and we started testing this in mice. And we're actually looking forward to going to humans relatively quickly, though. We had originally been planning on going into primates within the next year or so, and have some collaborators who think that we should go directly into humans and are going to look to do that in the next couple of years, hopefully.
It sounds as if this technology could have application across any medical problem at all. But are there some particular near-term candidates that are in your sights?
Yeah. The proof of concept was done in HIV, producing broadly neutralizing HIV antibodies, and these are things that have been studied for a long time, but no one's been able to effectively elicit them with a vaccine. And so we just tell the cells how to make them. Our first commercial application will likely be in the lysosomal storage disease space, doing enzyme replacement. And the reason we're after these is they're very rare diseases. There's a low regulatory bar to get into them, and in a very clear unmet medical need. So we'll go after them first. Then I think heart disease.
What would be a good example of one of these rare diseases?
MPS 1, multipolysaccharidosis type 1, and it's caused by a genetic defect that makes the patient unable to produce an enzyme called IDUA that they need to break down waste products in their cell. So it's typically lethal by around age 12, and your life can be extended by taking a replacement enzyme, but it costs over $250,000 a year, and usually only extends your life by about a year and a half.
And then you were thinking from those moving on to?
Probably HIV seems like a good fit, but has a harder regulatory bar. And the nice thing about it is it's very well supported, and there's lots of people who want to see it go into clinic. Heart disease is probably the largest application, and we would be producing more efficient versions of apolipoprotein, so good cholesterol basically that's better at pulling plaque out of your arterial walls.
So this could be preventive as well as curative.
Yeah, and the idea is that we produce something at a stable level for a long period of time. So we're not a great fit for something like insulin, for example, where you want varying levels and putting too much can kill you. You want something that should be relatively stable and ideally for a lifetime, so it's most effective that way.
This is, sounds to me virtually revolutionary. Are there kind of conceptual obstacles or misconceptions that kind of are problematic for you in trying to promote this new pathway, this new direction?
Yeah, I think early on, I mean, people thought the idea was fairly crazy. I mean, this was before Dendrian got Provenge approved. There were no approved cell therapies, and the traditional idea of gene therapy is modifying stem cells, and that didn't always go well. And so we fit in kind of an interesting space where we're taking relatively mature cells and we're modifying them and then putting them back, and they just produce their protein. They don't expand, don't take over the world. And you think of like some of the really fascinating work done with the chimeric antigen receptors, the T-cell receptor stuff for cancer. They're brilliant, but they completely go uncontrolled. The cells expand in the body, they obliterate everything they touch, and that's what they were built to do. But ours is it's kind of a unique way of doing this, and I think when we first started, it wasn't familiar, and now there's more interest in this space than I've ever seen, and I think it's actually a great time now conceptually.
What's next? What's the most important next step for you as you proceed?
I think getting the FDA on board with this for the clinical trial. I mean, of course, we're a startup, and so fundraising is always, I spend my life doing that. But we have, all the standard technical stuff of scaling up, and we're, collaborating with Hutch to do that. We have great partners for it, and but that'll be the next kind of scientific work going on, is how do you get this from mouse size to human size and go into GMP production. But yeah, I think the regulatory and just the general logistical hurdles of going into clinic is probably the biggest stuff on our horizon.
You mentioned the Hutch. Most everyone here, I'm sure this conference would know you refer to the Fred Hutchinson Cancer Research Center, but some folks out in vast television watcher land might need to know the full name. But it reminds me to ask you just about, what's it like being here in this community? Is this a good place for a venture like this?
I think it is. I mean, I'm still quite actively involved in the Bay Area as well, and have funding support down there, and core technology out of Caltech. But I think Seattle is it's a very friendly place. I found they have a huge wealth of academic and scientific resources, and they've been, I mean, we would never have made it to the point we did if it weren't for collaborators here who are willing to help us out, and often, for very little in return.
And so, do you find that to be true of this conference as well? Is this a good place for you to spend time?
Yeah, well, I've been going here. We actually, probably the first real public presentation we did was a poster session here, and so I've been coming for the last...
That was about how long ago?
I think that was 2009, I believe. It all blurs together after a while.
Yeah.
But yeah, so it's been great. It's exciting to see how many other companies are around here working on new ideas. And I mean, I've seen a few technologies I didn't know were existing around here, and so it's always stimulating and exciting.
Well, it sure is good to hear about the possibility of whole new approaches to helping people improve their own health with their little micro factories. I love that concept.
Thank you.
Thanks so much, and congratulations on your progress.
Thank you.
Thanks for being with us today. Thanks.